Hello and welcome to BioCode. In this video, we will see how we can utilize ensemble database for the retrieval of genomes or genome assemblies or data related to a particular species from ensemble database. So in this video, I'm going to utilize a species called polar bear, and we are going to retrieve its genome from the ensemble database. So just go back to the home page of ensemble, go to all species, and select your species of interest, polar bear in my case, and sorry, I selected the wrong one. So let's go back to this one, and from over here we need to select polar bear, and it should be about over here. There we go. We found the polar bear. You can also go back to the main page and view a full list of all species that are present on ensemble, and then you can retrieve information about that. So that is useful if you don't know what your species actually looks like, and if you have the common name as well. I'm going to go back, and from here I'm going to select the polar bear again, which is going to take some time to find it again. So let's see if it is available. There we go. So polar bear is as you know, it lives on the polar side of the earth, and this is the home page of a polar bear genome, and this is the scientific name, Orsus maximus, and you can see there is a category wise information which you can select. If, for example, if you would like, for example, you can do any sort of search in this particular genome. For example, if you have a transcript name, if you have gene name, variant name, phenotype, structure variation, somatic mutation, protein family, gene tree, genomic alignment, translation, protein domains, and that sort of information. You just need to select a particular category, then you need to select do any search. For example, let's search for GSK3b, and let's see if it has GSK3b, and then you will only see the result from this particular species, in this case, which is polar bear, and there is GSK3b present in polar bear. You can then go to the gene page and do gene analysis. But let's go back. This is the video on genome analysis. So basically there are 4 categories, right? The first one is genome assembly. This is the genome assembly accession, URSMAR, and 1.1, and the GCA. This genome assembly is basically accession ID from NCBI and or the ensemble ID, and then we can also download it. I will display show you how to do that. Then we have the genome annotation information, where we can download the FASTA files, GTF or GF33 annotation files. We also have the comparative genomics section, and we do have the variation if there are. Since this species does not have any variation database, we cannot really do that unless you have your own dataset. So first of all, let's see information and statistics about this species, and it will open up in a new page. We will find more information. The polar bear is a hyper carnivorous bear whose native range lies largely within the Arctic circle, encompassing the Arctic Ocean and seas surrounding land masses, that sort of information. Basically, this is a 3-line description of a particular species, particular organism. Then we have the assembly information. The assembly was submitted by BGI Shenzhen and on May 2014. The assembly is on scaffold level, so that means they had about 100,000 contacts, and then they were assembled into 20,000 scaffolds, and then they were converted into chromosome wise if there are chromosome available. Then gene annotation was also done, and as you can see, there is a gene annotation information available, that they carried out combination of protein to genome alignments, annotation mapping from all suitable reference species, and RNA sequence alignment, where they compared the RNA sequence data. So all of that information is available, how they did the gene annotation. Then we have the statistics as well. For example, assembly ID is provided. This will take directly you to EBI or MCBI. Then we have the base pair information, golden path length, annotation is provided by ensemble itself. Annotation was full gene build, so they actually constructed the gene build of the entire genome, Then the gene build information is also available, that it started on September and ended on December 2018, and gene build was last updated in March 2020. Database version is also available. Then we have the gene count. There are 18,724 genes available that are coding. Then we have the category wise information, for example, non-coding genes, and there are 5,296. Out of all of them, the category is done. And pseudogenes are also available. These genes do not have any function, and the gene transcripts are also available. That means these coding genes make up a total of 40,000 transcripts, and there are other GenScan predictions which are 48,000. So GenScan has its own parameters for the gene prediction, so the numbers will vary according to the genome that you are trying to analyze. So let's download this species genome, and let's click on download DNA sequence. It will open up a separate page where you will be able to download the sequences from a FTP link, and basically this FTP link will directly be able to will directly allow you to download the sequences from Ensemble database. So you need to you need to first understand if you would like the non-chromosomal region, or if you need the top level region, or that sort of information. So I'm just going to click on non-chromosomal, and since it is a scaffold level assembly, and the download will start, and it will take some time since it is 600 MB file. So let's go back to our main page. This is basically how you download the assembly. Then you can also you also have the ability to download genes, cDNAs, and non-coding RNAs, proteins, and other information. You also have the ability to download GFF3 file, GTF, or which basically means that this is annotation genome annotation files. So let's click on that, and this is how you can download the information. So let's click on GFF3, and it will download in a compressed files which is GZ, and it will take some time before it is finished. So in the meantime, we can go back to our CDS. So we can now retrieve CDS, DNA, which is the entire genome itself, non-coding RNA, peptide, which basically means the protein sequence. Let's open the pep folder, and where we will see the peptide or protein information. Let's click one of them, and we will be able to retrieve all of the proteins that are present within this genome. So let's download this one. In the meantime, we have now retrieved this information. I'm going to extract it on my desktop, and once it is done, I'm going to